In this video, we're going to be looking at the secondary messenger system in relation specifically to adrenaline and glucagon. I will cover it with adrenaline, but the principle will be the same for glucagon. So adrenaline is a hormone you heard of. It's secreted from the adrenal gland. You would come across it at GCSE. And we know it's responsible for the fight or flight response. So what does adrenaline do? Well, as you can see, there are three things that we think of in terms of adrenaline that I need you to be aware of. So the first one is it increases the heart rate, and the second one, it increases the breathing rate. The purpose of both of these, breathing rate and heart rate, is to take in more oxygen and take more oxygen to the cells. Okay, now you should know that more oxygen to the cells means that you can do more aerobic respiration. But the other reactant for aerobic respiration is glucose. So where does that come in? Well, adrenaline is also responsible for the conversion of glycogen to glucose in the same way that glucagon does. But obviously it's secreted in different conditions. So whereas glucagon is secreted when your blood sugar level drops, adrenaline is secreted in times of stress or fear or excitement. Now, why does it cause more glycogen to be converted into glucose? Because if you have more glucose and you have more oxygen, which we have now, we can now do more aerobic respiration. Which means where you can release more ATP, which means we can have more muscle contraction. Note the terminology more all the way, because obviously we're always doing aerobic respiration, we're always releasing ATP, but it's the fact that adrenaline will cause more of those processes, leading to ultimately to more muscle contraction. Okay, so the next thing for us to think about is how adrenaline actually works. So let's go back and think of something like a liver cell. So here is our plasma membrane of our liver cell. Now, obviously, our plasma membrane is made up of a phospholipid bilayer. And within those phospholipid bilayers, there are protein receptors. Those are the things that I'm interested in. So within here, there is a receptor, and that receptor has a very specific tertiary structure that makes it complementary to adrenaline and adrenaline alone. So that is our adrenaline receptor. Now, also attached to the bottom of the adrenaline receptor on the inside of my cell is another enzyme called Adenylate Cyclase. Now, at the moment, Adenylate Cyclase is in an inactive mode. Now, within my cell, obviously, I've got my nuclear membrane, which I'll draw in there, part of it. And within that, right inside, I've got my DNA, which has all my genes on it. Okay, I'll just draw one molecule of DNA. So we have adrenaline. So here is my adrenaline. It's got a complementary shape to my adrenaline receptor. So my adrenaline receptor will bind to my adrenaline, like so. And when it does that, it changes the tertiary structure of the adrenaline receptor. And because the adenylate cyclase is attached to it, it also changes the shape of the adenylate cyclase. And that change in shape makes the adenylate cyclase active. Okay, so let's write that first. So we have the adrenaline binds to its complementary adrenaline receptor. This changes the tertiary structure of the receptor, which causes the activation of adenylate cyclase. Now, what is the job of adenylate cyclase? Well, adenylate cyclase is responsible for the conversion of ATP to cyclic AMP, CAMP. Okay, cyclic adenosine monophosphate. So we've got ATP, we're converting it to cyclic AMP. This causes, catalyzes, we'll say instead, conversion of ATP to CAMP. And CAMP has its own responsibility. CAMP will also activate an enzyme. It will activate an enzyme which is called protein kinase. So it will take inactive protein kinase and convert it into active protein kinase. That's the job of the cyclic AMP. CAMP converts inactive kinase into active kinase. And then this protein kinase will then cause the transcription of genes required for glycogen hydrolysis. So the active protein kinase will then have to go into the actual nucleus and activate transcription. Now, what is important about this mechanism is that it is called a cascade mechanism. Now, why is it a cascade mechanism? Because one adrenaline molecule binding to one receptor will cause lots of cyclic AMP, which will lead eventually to lots of active protein kinase and therefore lots of transcription. So you are increasing the numbers from one adrenaline molecule, you're ending up with lots of cyclic AMP molecules. So it's a cascade, it's like increasing in number. Obviously, you need to know about the secondary messenger model. You're expected to understand it. It's also useful if you're writing an essay, for example, on the role of ATP, because you can talk about conversion to cyclic AMP, or if you were talking about nucleotide derivatives, because ATP and cyclic AMP obviously both come from nucleotides, because they both have the pentose sugar and the adenine and a phosphate or more. So, or you could talk about it in relation to shapes fitting together, because you have the hormone binding to the receptor. You could talk about it in an essay on enzymes. So there's quite a lot of essays in which this could fit in. So it's useful to be able to understand the process and to be able to understand why the cells do it.